Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Today's topic is applications of recombinant DNA technology. In this, you will be learning about different applications of recombinant DNA technology, mainly in the production of transgenic organisms like transgenic plants, transgenic animals, transgenic microbes, and how it is used in other fields like DNA fingerprinting, in disease diagnostic and gene therapy. Dear learners, let's see the different applications of recombinant DNA technology. First, coming to the transgenic microbes, that is the production of insulin. Insulin is the first recombinant protein produced for the treatment of diabetes mellitus to control elevated blood glucose level in the year 1982. Originally, this insulin was isolated from the fetal calf pancreas and other animal pancreatic tissue which is obtained from the slaughterhouses. So now it is commercially produced in a large scale in the recombinant E. coli cell by cloning the human insulin gene in the plasmid, and these plasmids, when you are introducing it into the E. coli cells are having the capacity to produce the insulin on a large scale. So in this manner, the human insulin or humulin, it is used for the treatment of diabetic patients. Second example is production of growth hormones. The human growth hormone or somatotropin is the main hormone which controls the growth, and the deficiency of this affects the nucleic acid synthesis, RBC production and fat metabolism. This growth hormone is produced on a large scale in recombinant E. coli cell in the year 1979 by Dr. Baxter's team. Coming to the next example, vaccine production. Recombinant hepatitis B vaccine, it is used to control the hepatitis B infection. It is developed using a recombinant strain of yeast, that is Saccharomyces cerevisiae, that produces this hepatitis B virus surface antigen, and it can be produced in bulk, and it is used as a vaccine to produce antibodies against the hepatitis B infection. If you see other example, interferons, these are the antiviral proteins which are produced for the treatment of various types of cancers and other type of viral diseases. They are produced in a large scale in recombinant E. coli cells. Coming to the second application, that is the production of transgenic plants. So you'll be studying about the insecticide resistant plants like BT brinjal, BT corn, BT cotton. This BT corn is resistant to European corn borer, whereas BT cotton, it is resistant to bollworm larvae. BT brinjal, it is resistant against this brinjal fruit and shoot borer. So all these BT plants, they are having the ability to produce an insecticidal resistant protein called as Cry1AB. So this protein, this is the toxin protein which is produced by Cry gene. This Cry gene is isolated from the bacteria called as Bacillus thuringiensis. Due to this origin, these plants are named as B and T. B means Bacillus, T means thuringiensis. Because the foreign gene is extracted from this bacteria, that is the transgenic plants are named as BT. Okay, so this particular insecticide resistant plants are commercially available in all countries, and in our country this BT brinjal further it awaits the commercial release, whereas the BT corn and BT cotton are in use. So this is the normal brinjal which is susceptible to this insect larvae, whereas this is the BT brinjal which is resistant against this insect larvae infection. Coming to the second example, golden rice. This is the normal rice and this is the golden rice, because due to this rice plant is having the it is a nutrient supplemented rice. It is rich in vitamin A. Due to this, they are appearing a slight yellow in color, so they are named as golden rice. So if you see the production of vitamin A in golden rice, the pathway it will be appearing in this manner. So this is the precursor, geranyl diphosphate. So when you are inserting two foreign genes in this normal rice plant, that is the one phytoin synthase gene, it is taken from daffodil, and the second gene, foreign gene, which is taken from Erwinia bacteria, it is phytoin double desaturase. When you are inserting these two foreign genes in the rice plant, they are having the ability to convert this precursor molecule into beta carotene. So beta carotene, it helps in the production of this vitamin A. In this manner, whenever you are inserting the foreign genes in the normal rice plant, it is having the ability to produce this vitamin A rich rice can be produced. Coming to the other example, plantibodies. So these plantibodies are the monoclonal antibodies which are produced in the plants. The plants which are used in this antibody production, they are tobacco, corn, potatoes, soybean, alfalfa and rice plants. And the transgenic potato, so you are able to produce antigens against this cholera and diarrhea pathogens, and even by taking this tobacco suspension culture, you will be able to generate the human monoclonal antibodies against this hepatitis B virus infection. That is, you will be able to produce hepatitis B virus surface antigen can be developed using the tobacco suspension cultures. Like this, various number of plantibodies can be produced in the plant cells for different types of human diseases like this cancer, dental caries, herpes simplex virus. Like this, different kinds of plantibodies can be produced in the plant. Antithrombin production. So this antithrombin, it is produced in the transgenic goat plants. This is the first therapeutic product which is developed by GTC Biotherapeutics, and it is approved by the US Food and Drug Administration in the year 2009. And this antithrombin, it is used for the treatment of deep vein thrombosis in patients who are suffering from the hereditary antithrombin deficiencies. So this particular recombinant protein, it can be produced whenever you are cloning the gene of this antithrombin in the embryos of this goat cells by using a viral vectors or non-viral vectors. The goat it is able to produce this protein, antithrombin protein, in its milk. So like this, different type of human therapeutic proteins, they are produced in transgenic animals. And if you take the transgenic sheep, alpha antitrypsin, then cystic fibrosis transmembrane conductance regulator, tissue plasminogen activator, and blood clotting factors 8 and 9, and fibrinogen. Like this, all these recombinant proteins, they are produced either in the milk or in the blood of this transgenic sheep, and they are used for the treatment of these diseases like emphysema, then cystic fibrosis, thrombosis, hemophilia, and wound healing. Okay, and if you see the transgenic pig, they are used for the production of tissue plasminogen activator and blood clotting factor 8 and 9. It is used for the treatment of thrombosis and hemophilia, whereas in goat, human protein C and antithrombin 3, they are produced in the milk or in the blood of this transgenic goat, and it is used for the treatment of this thrombosis. Like this, various types of this therapeutic proteins can be produced in the transgenic animals. And also the other application is the xenotransplantation of organs, that is the organ transplantation. So it is of much importance and it is useful to save lives. But the if you see the organ donation rate, it is very low. The frequency of organ donation, it is very low. And for successful organ transplantation, two things they are of much necessity. One is you should take care of graft rejection, and the organs which you are taking from the donor, it should be pathogen free or infection free. So these two factors, that is the graft rejection and the pathogen free, that is infection free organs, these two criteria are taken into consideration while transplanting these organs. So these transgenic pigs, they are now developed to achieve this reduced immunological rejection and better compatibility. So these transgenic pigs, they are taken as a source for the production of different types of human organs in its body, and they are used as a source of this organ transplantation. Next, coming to the other example, Enviro pig. So this that is, in this transgenic pigs, or they are developed by inserting a foreign gene called as phytase, which is isolated from the bacteria. When you are inserting this phytase gene in the pig, they are able to show effective feed utilization, and the fecal phosphate, the phosphate content in the fecus of these pigs, it is 75% less. Thereby it is reducing the environmental pollution, and it also helped in increase of milk and meat content, because due to effective feed utilization, the milk and meat content has also increased. So in this manner you are able to create this Enviro pig by using this transgenic technology. And next, so these are the Enviro pigs, or the transgenic pigs which are expressing the phytase gene. Now the trend has moved from gene modification to gene editing. This year's Nobel Prize in Chemistry, grabbed by these two scientists, Jennifer A. Doudna and Emmanuel Charpentier, for their work on molecular tools CRISPR-Cas9. The gene editing technique further gained momentum, and you will know the difference between gene modification and gene editing in the coming video.
Gene-edited food could be coming soon to a supermarket near you. What is it, and how is it different from GMOs? First, let's rewind. Farmers have been cultivating crops and animals for centuries through breeding. Then came genetically modified organisms. That's when genes are taken from one organism and inserted into another. Most of the corn and soy grown in the U.S. are GMOs. BT corn is a type of corn that has a gene from a bacterium that produces a protein that's toxic to certain insects, so that farmers don't have to spray as much pesticide. Most scientists say GMO foods are safe to eat, but some consumers aren't too sure. Gene editing is different. Instead of having to add foreign DNA, it is like molecular scissors that can tweak or remove an existing gene in a plant or animal to make food more nutritious and easier to produce. Take mushrooms that can go bad quickly. Researchers can cut out a DNA snippet that produces the enzyme that makes them brown. Scientists can also edit the DNA of dairy cows to make them hornless. They do this by snipping out part of a dairy cow's gene for growing horns and inserting DNA instructions from prized beef cattle that naturally lack horns. That would prevent injury and avoid a common but painful horn removal.
Yes, this gene editing technique definitely will rock the future of science and technology, right? Moving on to the other applications, gene therapy. Gene therapy is used to treat defective genes which causes the diseases. So here you will be using short DNA fragments as therapeutic molecules rather than the drugs. So in this gene therapy, you will be taking the DNA which is encoding a protein. The DNA is packed into a vector, normally a viral vector. So it's an inactivated viral vector which transports this DNA into the cells of the body. So this gene therapy was first used for the treatment of severe combined immunodeficiency syndrome. So this disease is caused due to the deficiency of the enzyme called as adenosine deaminase, and the SCID is the first disease which is treated by means of gene therapy in the year 1990 in United States. So if you see the picture, in this picture, so the cells, the stem cells, or they are taken from the patient and they are infected with this recombinant retrovirus. This recombinant virus, it is containing the gene which is coding the therapeutic protein. When these stem cells are infected with this virus, recombinant virus, the gene present in the virus, it will be transduced into these stem cells. Later, these stem cells are again re-injected into the patient. So in this manner, the particular disease can be cured by the insertion of this therapeutic genes. So in this way, the gene therapy is now in progress to cure different types of diseases. Later, coming to the next application in disease diagnostic, this particular technology is in the form of PCR or RT-PCR, that is reverse transcriptase polymerase chain reaction, and ELISA, that is the antibody test. This different kinds of infectious agents, that is bacterial and virus infections, can be easily diagnosed with these techniques, and it is also used for designing these RNA and DNA probes which are used to detect diseases such as cancer, foot and mouth disease, tuberculosis, muscular dystrophy, Huntington's disease. So by means of this PCR, the presence of the infectious agent in the blood sample can be detected in the form of the DNA band, and the absence of the band, it indicates a negative result. And also by using this RT-PCR, you can quantify the amount of the pathogen present in the sample, and the progression of the disease can also be detected by using this reverse transcriptase PCR. So it is this is the wonderful technique used for the diagnosis of this COVID-19. So in this manner, by using RT-PCR, you will be able to diagnose different kinds of diseases. Next, coming to the second application, DNA fingerprinting. So this technique, it is used to find out variation among the DNA sequences of individuals, and it is commonly it is used in forensic science for the identification of criminals, and the main concept behind is the VNTRs, that is the variable number tandem repeats. These are the stretches of the DNA which are which they will be differing from person to person, and this particular technique, it plays a very key role in identification of even the new species, and for evolutionary studies also this DNA fingerprinting, it plays a major role. So in this example, you will be seeing the DNA profile of two persons, person A and person B. Person A is having the size of this VNTRs is 12, that is he is having a 4 base pair sequence TC80 repeated 3 times, whereas person B is having the same sequence, 4 base pair sequence, but it is repeated 5 times. So in the first person A, the size is 12, and in the person B, the size is 20, because the sequence, the 4 base pair sequence is repeated 5 times. So if you run the sample of these two persons on a gel, you will be getting the fragments are eluted in a different manner. So in the person B, because of its big size, it is present in the upper portion of the gel, and the lower portion you will be seeing the person A sample because of its small size. So in this manner, you will be getting the DNA profile of the individuals or the organisms based on the size of this VNTRs. You will be able to identify the persons or organisms which will be having a unique profile. Next, coming to the other application, hybridoma technology. So in this method, antigen is injected into the mouse, and after few weeks you will be removing the spleen and extracting the plasma cells. So these plasma cells, when they are fused with the myeloma cells, so these myeloma cells, these are the cancer cells. When these two cells are fused, you will be getting hybridoma cells. hybridoma cells will be having the feature of this spleen cells, that is the production of antibodies, and the feature of this cancer cells, that is the ability to proliferate enormously, that is indefinite cell multiplication. So later when the cell these hybridoma cells, when they are that is induced into the mouse, they are having the capacity to produce monoclonal antibodies. So these monoclonal antibodies are highly specific for a particular antigen whichever you have injected into the mouse. That particular antibodies are produced and they are used for the treatment of different types of cancers and diseases. So in the last slide, let's recap the main points what we have covered in the previous slides. We have learned about the different applications of transgenic microbes, that is humulin production, human growth hormone production, vaccine productions, and how you are using transgenic plants and animals as bioreactors for the production of different kinds of recombinant proteins, and how this recombinant DNA technology is used in the field of forensic science in the form of DNA fingerprinting, the disease diagnostics, and in the curing of different types of diseases using gene therapy. And also we have learned about the difference between gene modification and gene editing. And with this I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.